Hello, everyone, and welcome to today's webinar, Integrating Spatial Resolution into Flow Cytometric Analysis. I am Michelle Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, visit thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Dr. Heaven Roberts, Protein and Cell Analysis Group at Thermo Fisher Scientific. Heaven, you may now begin your presentation.
Hi, thank you, Michelle, for the introduction. As Michelle mentioned, I'm Heaven Roberts, and I'm very proud today to be presenting about the Invitrogen Attune CytPix flow cytometer and how you can integrate that spatial resolution or imaging into your traditional flow cytometry analysis. So as I mentioned, I'm focusing today on the CytPix, and we can come back to this slide if there are questions, but just want to emphasize this is still an Attune. And so the Attune cytometer line uses acoustic focusing, and you'll see that we're capturing really very well focused images of a wide variety of cell types and across a wide range of run rates. And that is because of this acoustic focusing. As I mentioned, we image many cells, and sometimes we have a lot of fun with what we run through our CytPix instruments. So essentially anything you can put through a cytometer, you can image on the CytPix. So here we have some yeast, we have some sort of a spent necrotic immune cell with platelet aggregates. We have two species of algae or diatoms. And then we have a piece of debris that sometimes you'll see debris and you just start seeing things when you've been looking at images for far too long. I want to break today's presentation into kind of 4 segments. So really talking about problematic samples and rare events and how the CytPix can help with those samples. Also the unfortunate story of contaminant identification and how valuable the instruments become in our own R&D labs for this. For unknown samples or samples where you are unsure of the phenotype, whether you're measuring autofluorescence or you're measuring staining indices, this can really help you with gate definitions and with doing titrations and sample preparation. And this kind of leads into developing brand new flow protocols, maybe even of samples you've never worked with before, or samples you've worked with a lot and are just now discovering some new features. So first, let's talk about some problematic samples and rare events. These can be difficult to work with, and I know we've all had that question, what is this dot? The first thing we get asked of the CytPix by new users is, can you see doublets? And yes, you certainly can. So here we have several aggregates, and I've used our image backgating feature where I just click on the image and I backgate it onto any plot in my workspace. And I've put 4 potential aggregate exclusion plots. So the backgated aggregates that are doublets or triplets are in these orange dots. And then you can see the gray areas, the majority of my events. I only backgated about 50 events just to kind of show this as an example. But you can see here of the 4 candidate plots, really only side scatter area versus height is doing a great job at giving me a range that I can work in to draw a really good singlet gate. But even inside scatter area and height, you can see there's at least 1 orange dot down there right in the middle of where I would draw singlets. So if you are noticing that you're coming up with a lot of brand new conclusions on double phenotype cells, I highly encourage you to reconsider your doublet gating. First, I have found that using a very, very tight side scatter area versus height singlet gate is usually the best way to go. I've done this example on now hundreds of samples, and very consistently, this is the best gate for an aggregate exclusion. But also make sure that it's very tight up against that density. In addition, even though an event is well within those bounds, that doesn't mean it's not a doublet. So just always keep that in mind when you're doing your downstream gating. This is some data that a colleague of mine put together. He was looking at one of our new live dead viability dyes. Here it's the live dead fixable lime. And I think we've all seen somewhat of an intermediate population when we're looking at live dead plots. So sometimes that intermediate population is, as it is here, kind of shouldering off of that bright positive population. And we're pretty confident we want to choose just this leftmost negative peak. Other times, the intermediate population is shouldered against the negative population. So really titrating your viability dyes can be very important because there's been kind of contention for a while, at least from my understanding, on whether or not this intermediate population are dead cells or are these live cells that are starting to go into the steps of necrosis. So we can see here that that leftmost gate are our live healthy cells. Those are the ones we're after. The rightmost gate, of course, those are our dead cells. But the intermediate gate is also dead. In fact, they're so far dead that they've started to lose their membrane integrity. So for viability dyes that are staining nucleic acids, if the DNA is breaking down, or if they're staining proteins and the cell's leaking protein, this can really interfere with your staining separation for those viability dyes. So it's really nice to have the site picks to be able to do this quick gate check and make sure that I'm only including the live healthy cells I'm interested in. Here's a problem sample, we might call it, where we don't want to exclude dead cells and we don't want to exclude doublets, but we still want to get rid of debris. And we still want to learn more about the sample itself. So this is a CAR T cell killing assay where our target cells, our RAMO cells, are stained with the cell trace violet dye, and our CAR T cells are stained with cell trace far red. So we can see this beautiful quadrant plot, and we're able to determine which cell population is where. But the actual placement of those gates can vary quite a bit from sample to sample, and depending on your incubation times and the efficacy of the T cells that you've introduced the CAR. So it's important to be able to look at those images and see where the actual population cutoff is, what is debris, but also we can learn a lot about the timing and the methods behind how we measure these outcomes. So here we've just incubated for 1 hour. After an hour, what we were seeing is that the majority of the events in that Q2 quadrant are truly interacting. So we're having a true immune synapse. These aren't just two cells that are coming through the instrument together. You can see that here the CAR T is actually attaching to the RAMO cell and initiating killing. Moving on a bit to kind of a more problematic sample in a true sense. So this is an aged blood sample, and I'm sure we've all been in the position where, unfortunately, due to a centrifuge being down or a sample being delayed in shipment, we get our sample, we can't send it back. We certainly can't ask for a replicate sample. The day is gone. So let's make the best of what we have, and let's see what we can get out of this sample that still generates some good quality data. So here we've taken some blood that's about 36 hours old. We have done a centrifuge, a density gradient centrifugation to separate out our PBMCs, but we can see in this leftmost of these three large plots that we have a large contaminant population. We've called that R1. So we would assume that these are mostly residual granulocytes, probably mostly neutrophils, maybe some necrotic cells. In addition, in the next plot, we see this R3 population. So these would be low CD45 or intermediate CD45 expressing, hopefully, lymphocytes. I've always been trained that these cells should be gated out, but their actual placement can be a little tricky. So here they're pretty clearly separated from my main lymphocyte population, but that's in part because I've titrated this antibody specifically for this application. That's not always the case. So sometimes they're a bit more shouldered up to that main population and they can be a little harder to pull out. But some researchers have told me that they don't gate these out. They feel that they are a valid population to include in a downstream analysis, and that this kind of intermediate CD45 expression is of biological consequence and not an error and not because of necrosis. We also always have this lovely intermediate CD14 monocyte population. Some of these may be non-classical monocytes. They're an interesting population to look at, but we won't look at those today. I want to look at R1 and R3. So in R1, this high side scatter population, you can see we do have indeed some granulocytes and certainly a lot of cells that are going through necrosis and apoptosis. Some of them are very dead, and others are even still residual doublets, even without or even with a very tight singlet gate. For that R3 population, these are not cells that I would want to include in my downstream gating. So these cells are going through necrosis, apoptosis. You can see that some of them are shrinking and further advanced in that apoptotic pathway. Others have these apobodies floating with them or attached to them. It's a bit unclear to us if these apoptotic bodies are from this cell and they're being detached while it's going through the instrument, or are these floating in suspension because aged blood is going to put off a lot of apobodies. But it's still interesting to see these cells and kind of confirm that I don't want them in my downstream lymphocyte gates. This is kind of the full gating strategy for the same sample. And here I'm looking at NK-like T cells. So you can see I've removed my CD14s. I'm only looking at CD45 positive events in the lymphocyte gate. Um, this sample had numerous other gates associated with it, but this is the pathway we'll look at for now. And on its own, I would be very happy with this. I felt the titrations were appropriate, and you can see I've got a good separation of a small population, but that's to be expected. So what I can do now is look at the images of all the cells in that population, and at 0.17% of total events, I always have suspicions about rare event work, and now I can actually see whether or not these are real cells. Are they electronic noise? Are they just dead cells that are picking up stain that really doesn't belong to them? So they certainly are not electronic noise. And I will say that after looking at millions of images, I can tell you these look like NK-like T cells. This is the expected morphology. But you can see that a couple of them one has a spent platelet. We have an example here of one that has these two small apobodies. Again, this might have been picked up from another cell. So we could improve this gate. We aren't sure what that platelet or apobodies are carrying with them in terms of phenotype. And it would be concerning if we were trying to make a new discovery based on this gate, knowing that we have some contaminants. So here I've just redrawn that gate and I've called it repeat gate. So this is the same gate as the previous slide. And then I've redrawn it after removing every single problematic event. The problematic events here are just backgated in pink. And you can see that I've dramatically dropped down my percent of gated events, but now repeat gate 2 is a pure gate. This only contains NK-like T cells to the very best of my ability. So I think that the power of this is that it really gives you some evidence. And if you're submitting for publication, maybe you're submitting your gates, especially for those rare events, with these images to show here are the images that we identified that are non-target events. They represent a small percentage of our original gate, or even saying we've refined that gate based on these images, and now we have something like this, repeat gate 2, a pure gate. So now we're going to move on to kind of a sad story of contaminant identification. I think that many of us have experienced this, probably even more than realize, but contaminants do happen even in really well-controlled cell culture rooms. So this was actually the same assay on a different day, a CAR T cell Ramos killing assay. And we noticed, or a researcher noticed on a different instrument that they had kind of this altered forward and side scatter profile than what they were expecting. So we ran the samples on CytPix instrument, and we saw that even though these events were all staining positive for our Ramos stain, again, this happened to be cell trace violet, they certainly don't all look like Ramos cells. So in ROI3, kind of that low side scatter, that's where we would really expect our Ramos cells to be. And you can see they're there, but they seem to have some friends. In ROI1, with this high side scatter, these do not look like Ramos cells at all. In fact, this is what we suspect to be a fungal pathogen. Coincidentally, we had run the same cell line about a week earlier on CytPix instrument, so we were able to go back and look at that experiment. And we did see just a tiny fraction of events that looked like those ROI3 images. It looks like a fairly healthy Ramos cell, but it had these coincidence events that at the time we didn't notice or we kind of passed off as debris. But of course, now we're on the alert. So we bleach everything, we toss the cultures, and we think all is well. And unfortunately, several months later, we see the same contaminant, and this time in Jurkat cells. So a different cell line, these two cell lines are never interacting with each other, but they do share the same media. So we spoke with our cell culturist, and she mentioned, yeah, our Ramos cells we thawed last week didn't survive cryopreservation. This isn't entirely abnormal, so we just thought, well, maybe the thaw wasn't good, and had tossed that particular vial of cells, and she had started thawing another. We can see here the Jurkat cells are doing much better with this contaminant than the Ramos were, but we still have some of those really clustered pathogen events. So we decided to look at that new thaw of Ramos cells, and these are just 2 days post-thaw. And you can see some very early signs of very heavy contamination. It was interesting that these have a slightly altered morphology. They're a bit shorter than the more mature pathogen, but after we incubate them a bit longer, they do lengthen out, and it's the same species. I also like these images because the large cell, the Ramos cell, is actually triggering my side scatter, but because the small events are coming through the core at the same time, you can see how the core, the acoustic focusing, is really aligning all of these events. You can see it here in a two-dimensional space, but this is actually three-dimensional. And this is why we're able to get pretty well-focused images of something as large as a Ramos cell next to something as small as about a micron in diameter, because of that three-dimensional stability. And this is just another image. These were those Ramos cells a few days later. You can see these are very unhealthy, unhappy cells. But because the CytPix was able to pick up on these pathogens, now that we know what we're looking for, several days before something like trypan blue or just not seeing confluence, we're now able to put our cell cultures, we can run them off of plates using the CytKick, or we can even just quickly run them out of tubes. And we can kind of screen when we have new lines coming on or lines that we haven't worked with before, to make sure that we're keeping our cell culture incubators nice and clean. So now I want to move on to some more, for me, exciting, some unknown samples and new ways to do gate definition using the CytPix instrument. So here I have what we'll call a mixed culture sample or an environmental sample. And while I'm working with autofluorescence, you could use this same approach, and I have used this approach after staining cells, where I don't know which cells are going to take up which stain, but I do know that I'm hoping that my staining will separate my populations a bit. So here I'll add n by n plots to my workspace. So if I have 3 stains, I'll have, 3 by 3. Here I'm using autofluorescence, so I actually put all of them up, but I've just shown a couple as an example. I also, of course, looked at forward and side scatter, but you can see I just kind of have this continuous smear almost. That's really not very helpful in forward and side scatter, even if I change them to height or to log form. But it looks like red one, maybe blue one are going to be really valuable. So I've decided to keep these in this example. And what I'm going to do here is, instead of drawing a gate around these populations, or what I think are populations, and then looking at those images, is you can look at the images and you can just find cells that look similar, and you can backgate those cells onto your workspace, and it'll backgate onto all these plots at once. And so now I'm going to look at the fluorescence of each population instead of going the other way around. Here's just one example population. I know these are small and maybe look a little uninteresting, but to some, they're probably very valuable. But you can see they're very small forward and side scatter, and pretty low in their fluorescence as well. When we compare that to these, which are very dispersed in their forward side scatter and somewhat dispersed in red, they are, however, higher in autofluorescence than the previous cells. So using this, I came up with a gating strategy I can honestly say I would have never considered before. Here I have 3 areas of interest. I've called them OI 1, 2, and 3. They're on the top left. I actually decided to combine them. So these are things that I said, these are cells, and these are cells that I care about in my downstream research. To remove some very egregious aggregates, I've drawn what I've called a singlet enriched gate. I didn't remove all aggregates this way, but it's close, off of red 2, area and width. And then out of that, I've identified 4 major populations or important populations. They're certainly not major in their count. You can see that population 4 is only 0.39% of my gated events, but you'll see in a moment why this population is important. Population 3, I couldn't fully separate. I keep saying I'll come back to this and maybe someday I will, but I would like to stain these samples and run them again and try some different staining and separation protocols to better separate population 3. This is just those populations backgated onto a couple of those original plots, and you can see that they're not exactly where I thought they would be. So the continuous population in blue 1 and red 1, where I would have called that high population in both parameters a single population, is very much 2 distinct populations. Again, forward and side scatter is not very valuable. In this situation, I should say. And I said that I had identified 4. I actually stopped at 11. And from those 11, again, the singlet and the aggregate enriched, you can do this fairly quickly. And I usually backgate anywhere from 10 to 30 events, however many you feel you need to get a good consistent population. Sometimes you have them all land in the same spot. You only need 5 or 10. But you can really do a lot with this image backgating tool and really investigating where those events fall across these different fluorescent parameters. And I use this a lot, whether I'm working with algae, fungi, or immune cells. Here are those 4 populations, and you can see population 4, I mentioned earlier, it's very minor in its percentage, but this appears to be a dead population. So these are dead or dying, I believe, Chlorella vulgaris. All of these species are um, known for different sensitivities to toxins and to culture conditions. So if I'm applying culture conditions, or in my case, a toxin, to this culture, I really want to watch this population because this may indeed be the population I care the most about. And in this otherwise robust, healthy sample, it's very minor. But if that population grows, I want to know about it and as quickly as I can. Population 3 is actually 2 species, and one species has 2 forms. So you can see this on the left, you'll see 3A is the small form of the Chlorella zophingiensis, and on the right is this larger, darker form. It's much more autofluorescent. Every scientist always wants to talk about what they're doing next. So I'm excited now to talk about some new flow protocols I'm working on, and also what our software team is working on as well. So this is a sample that I had never worked with before and would have really not felt comfortable working with before using the CytPix instrument, and just starting to dive into this. So here I have a sample, and you'll see it here in a moment what that sample is, but um, I had an original idea of how I wanted to gate my debris. And I was just going to say that all of my really high side scatter and all of my really low side scatter was debris. This sample I have stained with cell trace violet. I'm trying to track these particular cells. So I was pretty comfortable that what I thought were my cells of interest are all staining intermediate to high with my cell trace violet dye. But if I just look at that population, after looking at the images, I can see there's still some debris. And so instead, I kind of went back to the drawing board and I started looking at those N by N plots, just as I showed in the previous example. And what I found was that I could do a lot better job of removing debris if I used, of all things, yellow 1. So by choosing just an intermediate population in my yellow 1 channel, these cells in particular separated out quite nicely there. If I look at that high population, which I've called R1, you can see that that has a very dispersed side scatter profile. And so really no side scatter gate that I could have drawn would have removed all of this debris. In fact, I'm only capturing about 75% of my debris if I use my original side scatter gate. So definitely use your autofluorescence to your advantage. It's not your enemy. It can be tricky to work with, but it can also be very valuable. These are the cells I was working with, and these are powdery mildew, a pathogen of hops. You can see that in my intermediate yellow and intermediate to high cell trace violet, I have these beautiful complete events. The reason they're intermediate to high is that there's actually several cells within this casing. However, in that high yellow one, I have these large aggregates of debris. There's still cells, there's still events, but I do want to remove them. And so as I'm working through my process to do gradient separation or improving my isolation protocol, this is the population I'm trying to get rid of, and R2 is the population I'm trying to keep. Once we start taking the next step and saying, well, instead of backgating my images, can I get data from those images? And this is what our software team has been working on. So we're looking at image processing that's brought back into the Attune software. So instead of looking at this in something like ImageJ, I'm able to look at this in line with all of my fluorescent parameters. And so this is just an example you can see on the left where I've taken something we're going to call entropy intensity. So, and I've put that against a live dead dye. And you can see that if I only had the live dead dye, these populations would not separate very well. I don't have very bright staining of my dead cells. But once I include that entropy intensity of the events, I can separate these populations quite well. This sample is differentiated to HL60 cells, and HL60s do differentiate on their own at a low percentage. But of course, differentiated cells are going to have a little bit lower skewness intensity, or if you think of skewness intensity in terms of roughness of the surface. So those undifferentiated cells are a little bit more erratic. They're ready to turn into anything we want. But our differentiated cells, which happen to be differentiating into neutrophils, they become smoother and they become much rounder than the original HL60s. So it was exciting when I started to dig into this and see that, this might actually be an earlier indicator of differentiation that I could then use to help me time my downstream work, which is neutrophil migration and phagocytosis assays. This also makes singlet counting even simpler. So earlier I showed you that maybe side scatter area versus height for honestly, the vast majority of mammalian cells is really the way to go. There's, coming soon is an even simpler way to do that, and that's something it's called spot count or particle count. And now I can just draw a very simple histogram gate, and you can see that these blue outlines are where it's saying there's a cell, as opposed to when I have aggregates, I have multiple cells, or in this case, cells with beads. Some of these other features are also very helpful in separating different types of cells, or again, cells and beads. Here I just have one population I've tagged as residual beads. These are human PBMCs that I stimulated with beads, and they were destined for a downstream debeading assay. So as I'm working through that debeading assay, I want to see this residual bead population disappear. And I want to see my healthy detached cell population remain intact. The unhealthy cells that are kind of that intermediate population, I'm not too worried about those. If I lose them in my debeading assay, they were already going through apoptosis or necrosis. And again, I can see that using some of these extracted parameters, as well as, of course, viability and an X and 5 staining. Here's another example of how this can really clean up a sample. So this is mouse whole lung homogenate prepared by a colleague of mine. And for those that have done mouse whole lung homogenate, it's not an easy matrix to work with. She's trying to preserve as many cells as possible. And so really trying to minimize sample preparation to the utmost degree. So it has a very simple staining process, and this is for essentially starting the process of titrating her antibodies. And so first thing we want to know is, are we titrating on live nucleated cells? And here, yes, we are. I mean, these are staining negative for a live dead dye. They are staining positive for a nucleic acid dye. She's prepared the proper FMOs, et cetera. And that does clean up this data substantially. But when we look at the images, we can see that While there are cells in these images, many of them are still attached to debris. Some of them have large blebs and other evidence of apoptosis. And we certainly don't want to go through the whole process of, sorry, an X and 5 folks, but an X and 5 staining adds another level of complexity to sample prep that can be a bit problematic when you're trying to do a lot of titrations on a very small number of cells. So another option is to use some of our extracted features to really clean up these cells. So here we're only looking at cells that have a fairly high intensity erythrocytes, because the images are illuminated off of a violet laser, will show up almost black from the heme. And so we can remove any residual red blood cells that survive the lysis by having a little bit higher normalized intensity as a threshold. Additionally, circularity, or the smoothness of the edges of that cell, will really reduce the amount of blebs and apoptotic cells that you include in your sample. So not only does this dramatically clean up the fluorescent plots with the nucleic acid and live dead staining, but you can see this really cleans up her scatter plots and just really makes this image view gallery look much more beautiful. These are the cells that we're after, and these are the cells that we want to titrate on. If we're titrating on cells that are dead or dying or have bits of aggregate, we're always going to find frustrations in those titrations, and ultimately we'll waste reagents and definitely waste time. This is just an example of kind of using the same approach and using it with the Cytkick autosampler. So because we can extract this data even off of Cytkick experiments, this was very exciting to start using this as a screening assay on 96 well plates. So these happen to be, these are Saccharomyces cerevisiae that I treated with carbendazim, which is a fungicide. And I treated them with varying levels of carbendazim, or I'll call it CBZ, for 18 hours. And what I noticed is that when I did this extraction is I got a much cleaner separation of my treatments using a combination of these extracted parameters along with traditional flow parameters. So here I've used side scatter and again the entropy intensity, and you can see that my diseased sample, which I'll call it here on the left, these are very shrunken, I presume nearly dead. Unfortunately, because these picked up every stain I threw at them, they made the staining profile on this nearly impossible. And so it was much easier and frankly much more robust to use these extracted parameters in combination with my flow parameters because of the altered staining profile of these diseased events. Here's my full titration curve. You can see that that disease state gate just disappears quite rapidly and is almost non-existent. It's 0.6% once we get down into the control, or the 0 micromolar. These are just, this is a heat map of our replications. So these are 6 biological replications. So each was pipetted separately for the carbendazim. And you can see it's extremely consistent using this gating strategy. I don't know that I've ever seen one of my titration curves at least go this well. So I was very happy to see how consistent these were and how robust that gating strategy was. And it really helped me to determine that really that effective dose for inducing this in Saccharomyces was somewhere between 400 and 800 micromolar. I might want to dial that in and repeat, but also hopefully kind of investigate further what's causing this. This is not the normal mechanism of action for carbendazim, to my knowledge. This is another example of how, in addition to offering a very straightforward and robust gating strategy, we can really show where our typical gating strategies have kind of fallen apart. So even for a very experienced user and using the image backgating, we certainly still make mistakes. And this is just one more tool to reduce those mistakes. So here I've looked at where I would have drawn a singlet gate as opposed to my singlet counting with these extracted features. And what I found is that I'm missing quite a few of my singlets. They would fall outside of my singlet gate. they're going to be much lower in their side scatter height versus area ratio. And so if I'm doing my best to exclude doublets, I'm removing singlets. Conversely, I'm including about 27% of my doublets in a singlet gate. So these are doublets that even with a fairly tight singlet gate, I just couldn't remove in traditional ways. So this is certainly very valuable for making sure that, especially if you have that rare event analysis where a doublet can make all the difference in the world, having this really robust singlet doublet discrimination is super valuable. This is an assay, I'm sure many of you used annexin-5 and propydium iodine, and having an intermediate population here is not uncommon. This can depend greatly on how long you've incubated your cells with your toxin, et cetera. These are Jurkat cells with camptothecin, and I use the dead cell apoptosis kit, so you can see my annexin-5 staining on this left plot is on my y-axis. This intermediate population here is about 15% of my singlets. And what was interesting in this is that I had the same user gating on 2 separate days. And you can see that where I have option 1 and option 2, I tend to draw these gates very similarly. I make little coffins for my dead cells and my apoptotic cells. And so at first glance, they look very similar. But even a very minor difference can pretty heavily influence your downstream outcomes. So if I'm trying to remove debris through manual gating, sometimes I can be inadvertently introducing error. And I think we've all seen this, and it's a known issue with flow cytometry, is that we have to be very robust and careful with our gating, especially when we go from sample to sample and day to day, where we will have mild variations because of biology or instrumentation or staining. So if we look at this compared to, oops, I apologize. Here, instead of using that kind of manual process of a side scatter and forward scatter gate, I've chosen to use something that we're calling pseudo diameter. So this is a derived diameter from the area. And so for me, this is a bit more applicable to the cells as well. I would expect apoptotic bodies to be very small in appearance, and I would expect healthy cells to be quite large. These are Jurkats. Keep in mind the pseudo diameter at this time is in pixels. So essentially my threshold here is for a 10 micrometer cell. Now, if I look at just my large events, you can see that I have quite a few that are annexin-5 positive. This is not surprising given how much camptothecin I gave them. And there we go. When I look at this in terms of replication, this was 3 sets of experiments. So the same cells, the same toxin, but prepared in 3 slightly different ways. And what I would hope to see is that one gating strategy emerges as the most robust gating strategy to use, even with these slight variations. And we can see that in terms of percent CV, my diameter has the lowest average CV across the 3 preparations and across my replicates. A CV is something that we can take and apply across multiple experiments as, in terms of a success rate of our gating strategies. That's why I chose to use this to compare these. But I think to me, this also plays back to, I can very easily explain to a new flow cytometrist or even a very experienced flow cytometrist why I chose a diameter threshold. And that diameter threshold can be adjusted for each cell type, but is consistent from day to day. Conversely, a staining threshold might be changed day to day depending on your titrations and your preparation, and of course your instrumentation. And so by integrating these derived parameters that really have a lot of biological impact, we can really start to improve the consistency of our gates and hopefully improve the consistency of our final research outcomes. With that, I do just want to thank everyone for their time, for the entire team that has helped make this instrument what it is, and also to allow me to speak today. With that, I will take any questions.
Thank you, Heaven, for your informative presentation. We'll now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. Okay, let's go ahead and get started. So our first question is, when using CytPix, do you change your flow rates to allow for better images?
Yeah, thank you. That's a good question. We can image at every flow rate, and I do not change my flow rates for images. We have some data on our website showing the same cells imaged at each flow rate. Um, I personally get a little nervous even just running at those high rates, but I think that's personal preference. I work with a lot of different cytometers, and so once you're past, 200 microliters a minute, I feel like you're screaming fast. Uh, but I adjust the instrument for my staining, for my cells, for all the other reasons we adjust flow rate first. And we always worry about the images last. We can take great images at each flow rate. So we definitely don't want to adjusting based on the images. The images come last after those adjustments are already made.
Okay, thank you. Your next question, how long does it take to train new users how to use the CytPix when compared to other cytometers or even the Attune NXT?
Yeah, so the CytPix is an Attune, so it's kind of the next generation of Attune. And for users of the NXT, I really think they're going to find it just really walk up simple to learn. The software is the same. We've added a couple of tabs, of course, for the image adjustment, but all the other features are identical between the NXT software and the CytPix software. They're actually one platform. For a brand new user, when I first started using the Attunes, I was surprised at how easy it was to kind of start learning the software. Um, so the same can be said for the CytPix software. We're able to train new users fairly quickly. We definitely spend much more time discussing, fundamentals of flow and why you might want to put a gate a certain way or make a voltage adjustment a certain way. The image adjustments themselves are very quick. And I want to say that if all you were worried about was image adjustment, you could probably train someone in under an hour easily, and that's a pretty long training session. I've shown lab mates in a few minutes and just said, yeah, here's your image tab, and here's how you'll adjust the focus and illumination, and here's how you know you've gone too far. Here's how you know you're really close. So it's really straightforward, and I think the software itself is pretty intuitive. Our team does a great job of making sure that it's very user friendly and that you can kind of find what you're after and you can move the panes around as you need. So compared to other cytometers, uh, some cytometers are great, easy, walk-up, user-friendly, and others, of course, take much more time to learn. So I would say that the CytPix is somewhere on that lower end in terms of the learning curve, but definitely feature-rich.
Okay, great, thank you. Looks like we have time for one more question. Why are some of the images a lighter background and others have a darker background? So, for example, on the slide with the dynabeads, the bead images have a light background.
Yeah, so at present, the software is actually scaling those images. So the images themselves are the same color. But it's scaling them to, think of it like scaling it to the darkest event. So because the dyna beads are so dark, because they absorb violet light, they're going to appear even darker on a lighter background. You can certainly adjust the intensity of the images in the software. You're not adjusting the image itself, just the display. And of course, you can take those images offline as well and do further adjustments to the brightness and contrast if you wish. If you feel like you acquired them and they're a little dark for your liking, you can brighten them up. We have found that sometimes when we're printing posters, it's good to make the images a little bit lighter because depending on the poster ink, they can end up being a little darker than you expected. But all of that is tunable in the software, and the images themselves have the same illumination across the entire sample.
All right. Well, thank you again, Heaven, for your time today and your wonderful presentation. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time.